Thank you for viewing this webinar on saving reports from the NextGENE Viewer. After creating a sequence alignment project with NextGENE, you can use the NextGENE Viewer to review the results and save reports. The report output can also be automated using the post-processing step of the project wizard or the Report and Export section of the NextGENE AutoRun. This first requires saving your report settings in the NextGENE Viewer. Several reports are available. We'll cover the features and settings for the Mutation Report, Expression Report, Coverage Curve, and Distribution Report. The Mutation Report is the primary report in the NextGENE Viewer, listing the variants for the project. The Mutation Report is shown either on the right side or at the bottom of the Viewer window. You can move the report display using the drop-down menu from the Switch Report Display icon. Click the Report Settings icon or go to Reports, Mutation Report Settings to open the Settings dialog for this report. In the Mutation Report Settings dialog, there are multiple tabs available. On the Display tab, you can select which columns will be included in the report. The Display tab is further broken up into sub-tabs. On the Annotation sub-tab, you can choose the columns with annotation information to be displayed, such as gene name, CDS number, and mutation calling nomenclature options. Click the Statistics sub-tab to view the columns of statistical information that can be displayed, such as the coverage, frequencies, and balance ratio. On the Phasing sub-tab, you can choose how to report phased variance. Phased variance can be called when there are multiple variants on the same reads within a few base pair region, such that a del-ints variant can be called. Requirements for phased variants are specified in the project wizard. You can use the Order sub-tab to modify the order of the columns in the report. Currently selected columns are shown in black. Click and drag any fields to modify the order. On the Filter tab, you can select options for filtering the report. On the Annotation sub-tab, you can filter based on the annotation. For instance, to filter out synonymous variants, or to filter based on the type of region, to include only variants in gene, exon, or CDS regions, plus or minus a set number of bases. On the Bias sub-tab, you can filter out variants that are directionally biased according to the balance ratio. The balance ratio is determined by the number or percentage of forward and reverse reads containing the variant. A perfectly balanced variant would have a balance ratio of 1, with lower ratios indicating more biased variants. These directionally biased variants can be more likely to be false positives depending on the chemistry used. You can select to use this filter and set the thresholds for filtering independently for SNPs, indels, and homopolymer indels. On the Score sub-tab, you can choose to filter based on the confidence score. This is a score calculated by NextGENE, including several components of the alignment at the variant position. You can filter by the overall score or any of the component scores. You can also select to have any of the component scores ignored for the calculation of the overall score. The Statistics sub-tab provides options for filtering either based on the same statistics used for variant calling in the project wizard or simply by a range of variant frequency. The values set here must be greater than those used for variant calling from the project wizard. For instance, if the mutation percentage threshold was originally set at 20%, setting a lower value here does not allow variants at less than 20% to be reported. The ROI sub-tab can be used to load a bed file for regions of interest for targeted sequencing. A hotspot VCF can also be loaded for filtering. On the Summary Report tab, you can choose how the mutation report will be shown in the Summary Report. The Summary Report is a customizable report that can combine multiple report types by loading the report settings files and can be saved as a PDF. You can enter a custom title for the Mutation Report portion of the Summary Report and select to include the full mutation report and/or Mutation Report Summary. You can find more information on the Summary Report in the webinar titled Using the Summary Report to Create a Customized Report. On the Output tab, you can specify additional formats for saving the mutation report when using the post-processing step of the project wizard. A tabbed limited text file and unfiltered VCF are saved automatically. You can also output consensus sequence FASTA files. The mutation report settings can be saved by clicking Settings, Save to File. The saved settings file can then be loaded in the Mutation Report Settings dialog for other projects. To have this and all other reports automatically exported according to the specified settings, you can load the settings files in the post-processing step of the project wizard or the Report and Export section of the AutoRun job editor. The mutation report can be saved by choosing Mutation Report from the Reports menu and then selecting the desired report format. All other reports can be opened using the Reports menu. The Coverage Curve report lists low coverage regions and can also provide a summary of coverage statistics for targeted regions. To open the Coverage Curve settings, click Settings, Settings. In the Settings dialog that opens, on the General tab, you can define the regions of interest and specify the coverage threshold for reporting low coverage. The regions of interest can be defined using a bed file or an amplicon text file. The bed file option is most commonly used and works best for alignment to a whole genome reference. Any regions within the targets where the coverage falls below the coverage threshold will be included in the report. Click the Display tab to select which columns will be included in the report. On the Summary Report tab, you can enter a custom title and select which parts of the report are to be included in the summary report. You can click Save Settings to save the settings file for this report. The coverage curve can be saved by clicking the Save icon or selecting Save Coverage Report from the File menu. Selecting Target Region Statistics from the File menu will display a report showing statistics relative to the target regions defined in the settings, including % of reads aligned to the target regions, minimum, maximum, and average coverage for the targets, and the % of the targets with a coverage greater than the coverage threshold specified in the settings. This report can be saved by clicking the Save icon here or going to File, Save Target Region Statistics. The expression report can be used to view the coverage levels for defined regions of the reference. After selecting the expression report, the settings are automatically displayed. On the General tab, you can specify the regions to be used. You can select regions defined in the reference annotation, for instance, to report for all genes or all coding regions. You can specify an incremental segment length, or you can define specific regions of interest in a bed file. Click the Display tab to select the columns to be included in the report. You can select which columns are to be included for annotation information and for the coverage values. On the Filters tab, you can set filters based on a range of RPKM or FPKM values, or you can exclude reads from the regions specified in a bed file. On the Summary Report tab, you can enter a custom name for the report and select to include the expression report and/or expression report summary in the summary report. Click Save Settings to save the settings file for the report. Click OK to generate the expression report. The expression report can be saved by clicking the Save icon or by choosing Save from the File menu. The distribution report displays a graphical display of the directional coverage distribution and can also be used to export the base-by-base coverage for selected regions. When the distribution report opens, the graphical display is shown. Each chart shows read counts by direction. Blue is used for the forward coverage, with red for the reverse coverage. The top chart shows the coverage distribution. The middle chart shows the distribution of the read starting positions. The bottom chart shows the read length distribution. Any of the charts can be removed by clicking the red X at the top right of the chart or using the View menu. By default, the entire reference range is shown. Go to File, Load Bed File to limit the display to the regions in a bed file. You can select Save Coverage from the File menu to export a tab-delimited text file with detailed coverage values for the defined regions. The General tab includes the options for this saved text file. You can manually enter a range of reference positions, or you can specify the regions using a region of interest text file or a bed file. Again, the bed file option is most commonly used and works best for alignment to a whole genome reference. For smaller references, you can also choose to save coverage for the entire reference range. You can also specify the step size for reporting the coverage. The default value is 1, which will report the coverage at every base position within the region specified. If using a value greater than 1, you can specify to report the coverage using an average or a sum for the number of bases in the step. The coverage table and coverage distribution graphics can also be included in the summary report. You can click the Summary Report tab to choose how this report is shown in the summary report. You can enter a custom name for the report and choose which sections of the report should be included. Click Save Settings to save the report settings file. Click OK to open a dialog where you can choose the name and location for saving the coverage table. Thank you for viewing this webinar on the NextGENE Viewer reports. Keep in mind, these reports can be viewed and saved from the viewer and can also be automatically exported when running new projects through the post-processing step of the project wizard and the Report and Export section of the AutoRun job editor. If you have any questions, please contact us at tech_support@softgenetics.com.